say, full organizing. Sorry for the invitation, and also all the that are in this moment online connected to my presentation. So the title is a little bit different, but again, so I will be discussing about the paper that was recently published in Nature Immunology. But also I want to essentially share with you our vision and the concept that is behind this idea of trying to explore this and exploit the space of non-canonical synthetic CD8 cells state for next generation cell therapy. So let me just 1 second. So there is no doubt that CD8 cells are key players of the immune system, specifically, you know, for controlling viral infections. It's very well documented, and we know it's well accepted, but also for controlling tumors. And in the last years, we have learned a lot, many things, new things about this particular population. And one of those things is the fact that this is not a homogeneous compartment, as we thought 20 years ago, that like, let's say, terminally differentiated killer cells. Indeed, they are more than that. They represent a continuum of T cell state that is a highly heterogeneous and with a specific T cell state with key specific functions in the context of acute or chronic viral infection, but also in cancer. And we have essentially learned many things about that, and also how to manipulate this, and also how immunosuppressant blockade can really alter the dynamics of this space. This is something that we have learned from systematically studying different tumor mouse models. For the reason here is one example in cancer, but also from studying different models of acute and viral infection in mouse. But also, we know that something similar is happening in patients with tumors. In this case, we can again see the same level of heterogeneity within the CD8 cell compartment here, specifically in human cells. So this is something that is very well accepted, and there are many labs that are quite involved in trying to understand these dynamics. And for sure, we want to do that, but specifically to improve adoptive cell therapy. Now, one of the most interesting observations that we have essentially made out of the literature is the fact that the number of CD8 cell state is much lower than expected. Here uses a summary of well-validated CD8 cell state from the literature that we can really classify based on the stemness potential and effector cytotoxic potential. Here I'm using the nomenclature that normally we use for mouse T cell, but it's going to be more or less similar for human CD8 cell states. And why I'm saying this? Because, for example, here we can, with this well-validated state, we can be around 10 different CD8 cell states, but only if we focus on one dimension. Sorry, because my mouse is not working. In one dimension, like for example, the transcriptional dimension of the CD8 cell differentiation, and we take into consideration, for example, n different transcription factors. Each of those transcription factors can be on or off. Theoretically speaking, we can end up with 2 to the n different states. This is a particular example. For example, we know from our data or analyzing, sorry, because I cannot access to the one anyway. So sorry, I will continue. I have a problem here, but I will continue. So here we know that, for example, this is an analysis that we have done, and we can identify in CD8 cells in the context of acute and chronic infection, we can identify around 1,000 differentially active transcription factors. This is very clear. So meaning, if you made the math, if you make the math, here we are talking about 2 to the 1,000 possible transcriptional states, but the system, in a way, is constrained to a small space of CD8 cell states. So we think that we are only looking at the tip of this iceberg, and more likely this has been the consequence of our interaction during the evolution with infection agents. Ideally, essentially a natural optimization process intended to maximize survival versus infection, but at the same time minimize death events due to immunopathology. So this is like that. We are quite interested to see if there is something here under the sea in this part, novel states that have been tested during the evolution, but we are super interested to see if we can really identify those states and clearly manipulate them. So saying that, I think that we can all agree in the concept that the space of CD8 cell state has been mostly shaped by this coevolution with infection agents. Because clearly this has been the more important selective pressure on the top of the immune system. And this has major implications for anti-tumor immunity, because we can see and we really understand, we can assume that the CD8 cell mediated immunity is constrained to the space of evolutionary selected CD8 cell states that has been indeed selected to fight infections, specifically chronic infection and not cancer. We know that most of the tumor-reactive TILs are indeed becoming exhausted. And this is a problem because we know that this is a program that has been evolutionarily selected to modulate the effector potential of these cells in order to minimize immunopathology. So meaning that we really believe that if we want to really push the frontier of cancer immunotherapy, specifically thinking the next generation of ACT and cancer immunotherapy in general, we need to really move far, far away from this heavy object and trying to see if we can illuminate and explore new regions of this space. And this is essentially what we are doing in our lab, specifically under the assumption that the space of CD8 cell state can be precisely defined by these three functional dimensions: the transcriptional dimension, the metabolic dimension, and the epigenetic dimension. And under that assumption, so the question is if we can really identify non-canonical T cell states that are in this space, specifically outside of the region that has been essentially occupied by the canonical exhaustion program, and if some of those states are endowed with better anti-tumor effector potential. And we want to characterize those states and see if we can also push a bit more in order to really use this basic information to improve cancer immunotherapy efficacy in the clinic, but also specifically adoptive cell transfer. So here, today, I want to talk to you about the idea that is essentially the idea that we cover in the paper, is that in contrast to immunocheckpoint blockade mediated revigoration that is essentially taking place in the evolutionary constraint space of the cell state, because this therapy is not able to really induce novel states, orthogonal cytokine engineer that is defined by a simple concept: engineer T cells to express factors to promote steadiness and factors to promote effector differentiation. And with this particular sample combining a specific variant of IL-2 with IL-33 to promote inflammation, we were able to see optimal tumor control. And this optimal tumor control and good tumor rejection was indeed associated with the generation of a particular differentiation pathway that was completely orthogonal to the canonical exhaustion program. And this program, specifically the effector state, has potent anti-tumor effector functions, specifically expression of several effector molecules, such as those molecules like granzymes and perforin. So I want to start with, we started this project 4 or 5 years ago with this question. So we wanted to really see and improve the efficacy of ACT in the absence of lymphodepletion. So I know that you should know, I'm pretty sure that you know that lymphodepletion is a very important characteristic, a very important feature of adoptive cell transfer for all the modalities, and essentially is there. It can be done with total body radiation or with chemotherapy, and specifically is there to create a space for the cell that we are transferring. Here on the right, you can see a very particular example of adoptive cell transfer, in this case, in the context of visiting over tumor models. When we transfer between 5 and 10 million of 41 cells, which are tumor specific, and you see that without lymphodepletion, there is essentially no significant tumor control. This is not, the control cannot be improved even though you add on top of this, for example, PD-1 blockade, as is shown here. And this is something that we know has been characterized, quite well associated with the fact that the persistence under this condition is very low. So for that reason, that was essentially the first problem. How do we improve the persistence of CD8 T cells within the tumor, specifically intratumoral persistence, intratumoral engraftments is the same thing, in the absence of lymphodepletion, also in the absence of cytokine support. And for this, we essentially use a molecule that we brought from Cuba. It's a variant of IL-2 that was designed there, specifically in the Center of Molecular Immunology, and was rationally designed to not to bind the IL-2 receptor alpha, which is CD25. At that point, essentially was intending to relax a bit the competition between conventional cells and regulatory T cells. But here in the lab, you can access to this observation in bioRxiv, we have demonstrated that a very important characteristic of this molecule is that increase the stemness potential of CD8 T cells in comparison with wild-type IL-2. So we did experiment, we transferred those cells without preconditioning in advanced B16 over models bigger than 100 cubic millimeters. However, in those experiments, we never saw effective tumor control. However, we learned two lessons. The first one is that even though you don't see tumor control, the persistence can be improved, as you can see here, without any external manipulation. And more interestingly, also we learned that these cells, the tumor-specific cells that will persist in there, were mostly tox negative. You know that tox is the transcription factor that is indeed the master regulator of exhaustion. And one putative problem of this first intervention was the fact that in this context, the cell will persisting, but will persisting as TCF1 positive cells. So meaning that like stain-like cells indeed was a very important issue of differentiation, and then you need effector cell to promote tumor rejection. This is something that has been quite demonstrated. We know a bit more about this state because also in the paper we show that with this perturbation, we are also able to induce a non-canonical T cell state that we do not fully understand right now, but it's like a memory, a hybrid state with feature memories and also effector-like features. But these cells are completely, as you can see here, out of the exhaustion program. At the gene level expression, and also we validated many of those markers by FACS, we can see essentially upregulation of CD25, activation marker RC69 or 41BB, and also the upregulation of this stain-like marker like CCR7. But remember, this state is not really good in rejecting tumors. So in order to fix this problem, so we realized that, as I said before, we need more differentiation. And for that, we bring to the scenario IL-33 secreting cells, because it has been demonstrated in many other papers that it was demonstrated that when you give systemically IL-33, this intervention is associated with tumor control and specifically with inflammation within the microenvironment. So we essentially define this very simple combination, and as a common denominator, we also engineer the cells to secrete a PD-1 decoy. This is a molecule that is essentially formed using the ectodomain of the PD-1 fused to an IgG4-Fc, and is essentially to block the interaction between PD-1 and PD-L1. Now we know that it's not necessary, and in the paper we show that. I'm coming soon to see essentially to show you that, but clearly once those cells acquire non-canonical, non-exhausted states, even though PD-1 is there, they are completely refractory to PD-1 mediated inhibition. So now in this setting, we just essentially came back to our model with, in this case, with B16 over tumor cells for the proof of concept. We also demonstrated the preclinical efficacy of this therapy with other tumor models, NC30, and also with other TCRs like PMAIL, but specifically for this proof of concept with OT1 transferred 2 days, as you can see here, without preconditioning, without cytokine support, and then we essentially follow up the mice. And here you can clearly see that with this perturbation, we could see significant tumor regression. We also in the paper are showing what are the reasons of the escape, specifically loss of the antigen by downregulating the MSC class molecule, as expected, but in this setting was quite evident. And as you can see here, for the first time, we were able to see that in this setting, the cells were persistent, and also we could see that there was an effective process of effector differentiation based on the number of TCF1 negative cells, effector-like cells, that we observed during the control. After this, so we came with a question: does orthogonal cytokine engineering induce non-canonical CD8 cell states? And to tackle this question, we essentially were looking at these different dimensions. That at additional level, that is essentially showing the paper, but also we'll share with you some new data about the epigenetic and the metabolic states that will be part of a paper that we are essentially also that is in preparation. So for this, we essentially did single cell RNA-seq in different time points after the cell transfer. Those that are in red are the time points associated with the response. And we also sorted independently the engineered cells and also the endogenous cells. And when we analyzed the data, we realized that during the response, most of the transfer cells were not distributed along the same axis of state than the endogenous. We know, and we annotated those states based on several markers, like the canonical state from naive to terminal exhausted. And as you can see here, the transfer cells were concentrated, were focused specifically in the cluster C5. And as I said before, during the control phase. And also we show in the paper that during the escape, this state is also lost, clearly showing a very clear association between the presence of the state and the tumor control. So if you go inside this, you can clearly see that this is an effector-like state based on the lack of expression of TCS7. And here you can clearly, again, see that these are expressing PD-1 based on the expression of the PDCD1 gene. And as clearly seen here, they are completely tox negative. But that is not only tox. For example, if you look by this at the expression of others transcription factors that are important for the exhaustion process, you can clearly see that factors like NR4A1 and NR4A2, which are critical for this, are also downregulated. Similarly, we observe the same thing with NFAC1, NFAC3, BAF, BHLHA40. So clearly, at the transcription level, by the expression level, the state also look different, not only because they will not express in tox, but also they will downregulating other transcription factors that are critical for exhaustion. We know also now, but I will not show that also this state, if we infer the transcription factor activity, we can clearly see important differences, specific differences in terms of activity of several non-canonical transcription factors in these cells relative to exhausted cells. But it's not only at the level of transcription factors, if you here are comparing with the terminal exhausted cells, but also if we look at other genes, like for example, granzymes, we see that these cells are expressing a set of non-canonical granzymes. You know that the terminal exhausted cells mostly express granzyme A, granzyme B, and also the exhausted intermediate state cannot regulate those granzymes, but they never, ever express the whole family of granzymes. And for example, here, granzyme C is one of those are quite specific of those states, and we can track this population by FACS using antibodies specific to granzyme C. We also in the paper did a very, we compare our the transcriptome data of our single set of our T-cell state with almost all the T-cell states that have been reported in the literature, just to demonstrate that that was essentially very unique CD8 T-cell state. Here is just one example of the comparison with what Laura Kodari described as better effector cell and was published in Nature. It's essentially a very similar approach using, again, a non-C25 IL-2 variant. As you can see here, the effector state that they are describing in that paper also downregulate tox. But when we directly compare those states, even though they are projecting the same region, the better effector cell projecting the same region that our C5 that we call synthetic effector cells, just because of that, because they are unique, we can clearly see that there are many important differences in terms of gene expression. And again, so we can see that the lowest expression of tox is observed in our state in comparison with this one that again is associated with better tumor control. In the paper, you can also find extra comparisons and also with T cell state that have been observed in the context of viral infection, acute and chronic viral infection. So we also, as I said before, we're able to formally demonstrate the existence of that state by flow cytometry. Specifically, we use antibodies against granzyme C to detect this state. And you can see that during the response, we can clearly see that there is a majority, that the majority of cells are granzyme C positive, TCF1 negative, which is essentially suggesting that this is the state that we saw by single cell RNA-seq. We also observe a population that is still expressing TCF1. Also in the paper, we think, well, we discuss and we think that they are essentially the precursor of this population. We are in our way to formally demonstrate that. And as you can see here, so if you use essentially CD44 positive, PD1 positive cells, stills from non-treating tumors, just as control of the exhausted compartment, because some of them will be exhausted, you can clearly see that PD1 positive, tox positive exhausted cells barely express granzyme C. So this is a very specific marker that is clearly associated with this differentiation pathway. It's not quite expressed in the context of canonical evolutionary selective CD8 T cell states. We also in the paper show that that was quite specific of the combination. You need both of them to induce that state. In this single cell RNA-seq, we did bulk TILs, but we could see essentially the fraction here. Also, we had endogenous. And also we validated by facts that most of the cells in the context of the combination will acquiring this state. So you need essentially both things. And also we were able to demonstrate that these are polyfunctional cells. And the polyfunctionality is associated with the expression of granzyme C, which is essentially the marker that we are defending for this state, as you can clearly see here. And also, if you isolate those cells and then you transfer those cells together with tumor cells to a new mice, we can clearly see that there is a very significant delay in the tumor engraftment only when you combine the tumor cells with the TILs isolated from the orthogonal therapy, and it's clearly much better in comparison with tox-positive exhausted cells that in this case were generated in vitro, because essentially we had a problem with the number of cells that we can isolate exhausted cells because they do not persist a lot. And also a very interesting thing that we were able to formally demonstrate in the paper was that although these cells are PD1 positive, these synthetic effector cells are not functionally restrained by PD1. To demonstrate that, what we did was to sort the cells again at day 12, and as control we use activated naive, and we expose those cells to TCR stimulation with or without PD-L1. And while the activation, the proliferation, the expression of several cytotoxic molecules can be affected in the context of these cells that are activated in a natural way for 48 hours during in vitro, we never saw essentially that the cells isolating from the tumors were suffering for having the PD-L1 FC in the culture. So we essentially don't know why this is happening, but our hypothesis is that the pathway, as soon as you have a completely different wiring, the connection between PD-1 and the intracellular pathways are somehow lost as a consequence of this reprogramming. This is something that also we are trying to validate. So now I want to share with you new data that is part of the work of a new PhD postdoc in the group that is essentially associated with the fact that this is not only these are not only transcriptional differences but also epigenetic differences. We did indeed single cell ATAC-seq, and it was very similar. So we essentially did the ACT and we collected the cells, the OT1 TILs, the engineered one, in different time points. And also as control, we sorted CD44 positive cells, CD8 cells, from non-treated mice. This is what we call endogenous TILs, when we will have a representation of both effector memory and exhausted cells. And also as control, we sorted splenocyte, specifically naive OT1 T cells, and as you can see here, we did single cell ATAC-seq. And here you can clearly again see that the transfer TILs, the synthetic TILs, are also clustering in a different region of the space than endogenous TILs. And also, over time, we can clearly see that a particular population, here characterized by the particular epigenetic profile, is also coming with the same kinetic that we've observed when we're characterizing this state at the transcriptional level. If you go deeply in this epigenetic profile, we can clearly see that this new cluster is essentially compatible with the clusters that we have seen at the transcriptional level, with a specific upregulation of several granzymes. Just here, I'm showing granzyme C again and granzyme A, and here you can clearly see that the accessibility for several exhaustion-related markers, again, TOX as a master regulator, is significantly reduced in comparison with a cluster that is representing the exhausted state. Here, you can also see, specifically here, looking at the enhancer of TOX, which is essentially the one, is the part of the gene that is directly regulated by MFAXI1 and MFAXI2. You can clearly see that it's completely close in the synthetic effector cells in comparison with canonical exhausted cells. And again, granzyme C, this locus is more accessible in the synthetic relative to the exhausted cells. But not also granzyme C. This is something that we have systematically seen in this context, is that one important characteristic of this novel biology is that these cells open the full loci that is containing the whole family of granzymes. You can clearly see here that in the context of canonical exhaustion, this is restricted to essentially granzyme B. We know that TX expresses granzyme B. But here you can clearly see that the availability of granzymes to kill is essentially quite optimal, or is enhanced in the context of our synthetic cells. This is just to close this part. Also here, you can now see all the differentially accessible peaks across this state. And particularly when we analyze the enrichment of transcription factor binding motif, we can clearly see that in the synthetic effector cells, the accessibility to the family of transcription factors that has been associated with canonical exhaustion, like the family of NR4A1, interferon-related transcription factor, or MFAP, is completely lost in comparison with essentially the level of accessibility or the enrichment of this motif in the cluster that is representing the canonical cells. And also, now we know that this state, specifically the synthetic effector state, is quite enriched in transcription factor binding motif that belong to the BC family of transcription factor AP1. But the most important thing for us is that in this, somehow, when these cells are going to this differentiation program, they are less permissive to all the transcription factors that are induced upon chronic TCR stimulation. So meaning that we think that these cells are, they can really allocate a high level of TCR signaling with, and also this transcription factor can be highly active, but they will not do anything because they will not find the region in the genome to regulate. This is something, to illustrate that indeed. So we have now validated that these are essentially different epigenetic states. And just to finalize my presentation, I want to show you a couple of things related with the metabolic dimension, just to really, go with the concept that we really believe that we are we are out of the region that represents in this space the canonical exhausted states, is that also in the paper we show that in the synthetic TILs, we can see many metabolic related pathways that are preferentially enriched in comparison with TX. For example, you can see here pathways associated with ATP synthesis or production of energy or with biosynthetic processes, many of them. And also here in the right part, you can see, for example, that the synthetic effector TILs are enriched in the signature that is observed in TILs with high mitochondrial potential, but also is enriched in some processes associated with mitochondrial metabolism. So we really think, and we are working on that direction, that indeed there is a completely novel and very interesting metabolic reprogramming. We also have validated, Joanna, in this case, a postdoc in the lab, has started with the validation of many of these kind of things in order to also made the point about the metabolisms, and we indeed have seen that in the in this case, a direct comparison between our synthetic cells, sorted cells, with sorted, in this case now OT1 exhausted cells, we can see a trend of having higher mitochondrial mass, but it's obvious, and also as we saw in the GC analysis, what is very interesting here is that the synthetic TILs have better mitochondrial, higher mitochondrial potential than the exhausted, and overall better mitochondrial activity estimated using the mitochondrial potential and mitochondrial mass ratio. So we are essentially also working on that direction to formally demonstrate that these cells are indeed different at the metabolic level. So now, to finalize my presentation, I hope I could convince you that we are essentially in our way to formally demonstrate that those states that we are observing here are quite outside of the region that we expect to see the canonical exhausted cells. So far, we know that this is a process and we are working on that, but just to let you know that we know that this is something that has somehow associated with two factors. The first one is essentially the in vitro expansion. This is something that is important for this process. And then also the in vivo manipulation, the fact that these cells are continuously secreting mostly IL-2 variant, which is critical for this phenomenon in the context of tumor inflammation, not only with IL-33. IL-33 can be replaced by other molecules, and we can get specific molecules that induce inflammation, like CD40 agonist or CD40 ligand, or in combination with radiotherapy to induce inflammation, and we can induce tumor regression, clearly also induce similar non-canonical T-cell states, all of them in this moment having as a common denominator this continuous secretion of IL-2, which is IL-2 variant in this case, what we think is essentially one of the most important things, and now have been validated in some studies, and hopefully in the last one, too, from the Wells lab. And here also, we are now quite interested in go beyond that, specifically trying to demonstrate that we have similar phenotypes with human T cells, and most importantly, trying to generate a lot of experimental and computational strategies to do what we call laboratory evolution of T states in order to see if we can really customize the cell states completely outside of the exhaustion program, and see if those are compatible with full tumor rejection of poor immunogenic tumors without preconditioning, without cytokine support, and we minimizing the level of side effects if possible, because this is something that we can really also minimize by just removing the preconditioning regimes. So with that, I'd like to thank Professor Joe Kukus, and also for the possibility of working with him, and also now for setting up this new program together with him, and also with the members of our lab, specifically to Joanna and Tanya, because essentially they were quite important for the second part. I'd like to thank to our collaborators, Santiago Carmona, which is the group leader of the Cancer System Immunology Lab from UNIL, and also Massimo Andriata. From Cuba, our collaborator, Dr. Khalil Leon, that was essentially the guide behind the IL-2 variant, the design there and the proof of concept there for a different purpose. but essentially, and also for the time that we essentially spent together discussing about this, in the past. So now I'm done. Thank you so much for your attention, and I will be extremely happy to take your questions.
Thank you very much, Jesus. That was really amazing presentation and amazing science that you're doing. So yeah, if anyone in the audience have any questions, I will take the first one. That would be, so this production of these cytokines is constantly happening. So it's not a transitory production of the cytokines. Are there? My question is, what happened if this novel state, then you take out these cytokines? What happened to these T cells that are becoming like sort of memory cells, or what happened if you take that stimuli after the tumor burden?
No, we know that the production for somehow is quite constant, and you also can see this dynamic in the paper that is in bioRxiv. Is quite constant during the response. For some reason, also at some point during the escape, also the escape is associated with loss of IL-33, but not a lot of IL-2 variant. And in that case, what you get is essentially this hybrid state that we call the C7 memory-like state that is essentially driven by IL-2. So this is what happens if you completely withdraw 33. In that setting, we cannot really manipulate and completely eliminate the secretion of IL-2 variant, but clearly, because it's continuously secreted. But this is a very nice question that we will know, because in the clinic, so we are taking the IL-2 variant to the clinic, and it's going to be under an inducible promoter. So clearly the cell will not be producing IL-2 variant or CD4 T ligand all the time, and then we will see. But yes, without 33, there is escape, and they become this kind of hybrid memory-like state that is associated with IL-2 variant.
Okay, and my second question would be, I was looking at some other publication that actually in acute model of infection, IL-33 is really important for the CD8 T cells itself. And your data says that in the tumor microenvironment, you don't have the CD8 T cells are not getting activated by ST2. What are your comments on that particular condition where in acute infection you require ST2, but or IL-33 signal in the CD8 T cell, but not in a chronic state of inflammation?
We're tackling that question, and for some reason, and also in it's shown in the paper. Specifically, the cells that we are expanding in vitro, they are not really expressing ST2. So and also we never detected ST2 by RNA level in the single cell data, but when we did the staining using a well-validated protocol from one of our collaborators, so we didn't see expression of ST2. So it's important, if you remember, our concept is that these cells are expanding in vitro and they are manipulated with IL-2 agonists, and then they are secreting IL-2. They are not behaving like the cells that you are getting there in the tumor, or the cells that you have in chronic infection that are the classical PX and TX that are expressing ST2. We know that ST2 is expressing the myeloid compartment for sure, because we have done single cell RNA-seq data from the myeloid compartment, and we can see cells are really expressing ST2, so they are sensing ST2, but not in our cells. And also here it's important that we are also altering the biology. IL-33 is never secreted. So it's an alarming that in the natural biology is never secreted. But here we essentially force the cells to secrete IL-33 to the environment. In my opinion, because it's continuously secreted, this is a very active promoter, the one that we have in the vector. So the levels, I don't think that are physiological. You see? For example, if you go to some of those papers about 33, you will see that 33 is important for maintaining the precursor receptor light population. But here we don't see that. We essentially see that when you have secreted 33 by these cells, you promote more effector differentiation, more TCF1 negative effector differentiation. So something is, and again, you can replace 33 by other stuff, like, for example, anti-CD40 to induce inflammation, and you get tumor control, and you get similar states. So 33 is just one example.
Okay.
You can do radiotherapy, but what is important here is to keep IL-2 variant and induce inflammation in microenvironment.
Thank you, Jesus. So we have a question from Daniel. I will unmute yourself.
Oh, okay, hi, I can start my
Hi.
video. Very nice talk, Jesus. I have a couple of questions, if I may. So the first one would be, so I like a lot your introduction about all these different states. Now I can start my video. So all these different states that you mentioned and how they have been constricted by infection or cancer. And I was wondering if, if you not use, as you say, the IL-33 that is normally doing the tumors and so on. So how is your thinking for designing these combinations of cytokines to get to a certain states that you would like to get? Because it's more like you take from the literature and then you say, okay, this cytokine may be good for the function that I would like to get, or it's more like testing all different combinations and see which were better for the phenotype that you need?
Perfect. So this is a great question. I have to say that when we started this project, we were not thinking about novel T cell state. We were thinking about the theory, but we didn't have any issue to connect IL-2 variant with this. So we were using essentially IL-2 variant because, you know, IL-2 is super important in the context of cancer immunotherapy since the 80s. So in this, now we had the opportunity to play with this molecule specifically for stemness. But we were not pretending. We had the theory at that point, but we were not pretending. We didn't have any rational inspiration to say, okay, this is for that. This is, to be honest. So the rational inspiration was a cytokine for stemness and a cytokine for inflammation in order to, you know, to fight or to completely work in the assets for conditioning. Afterwards, we realized that something different is happening. And now we are essentially connecting this with essentially this orthogonal path. This is essentially afterwards. At the beginning, no clue about that. You know, we had always the inspiration. We will, in my opinion, because, you know, this is something that now we know is connected with IL-2 receptor signaling, in this case, IL-2 variant. And we know that the IL-2 receptor signaling is able to counteract chronic TCR stimulation, is able to go directly within the tox locus and block tox. This is something that now we know, and now we are exploiting this. But at the beginning, no, for sure.
Mm-hmm. And if I may, a second question. So I don't know if I missed it from the paper, but is this secretion mostly acting like autocrine, so to the same cell, or is helping the other cells that are around?
Yeah, it's a very good. It's a good question. You know, you will not find it in the paper because we didn't have space. But this in the bioRxiv paper. So we in the paper you will see in the bioRxiv paper, you will see that you know, when we analyze, because by single cell RNA-seq we analyze the endogenous compartment, and we essentially look at the endogenous PX and PX, and we can clearly see that there is down regulation, you know, of the exhaustion program in the tumor, in the putative tumor-specific cell in the endogenous. They are down regulating tox as well. They are acquiring a transcriptional profile which is similar to the same. It's not completely the same profile, but it's very similar. Down regulating tox, up regulating granzyme. We also verify this, and also we played with the chimera model when, for example, we can have the OT1 cells as part of the endogenous compartment, and then use a congenic OT1 secreting the molecules. So meaning that you can, you know, clearly distinguish between endogenous OT1 or endogenous OT3, for example. We did it, can find in the bioRxiv paper, and we can clearly show that in this, during the response, the endogenous previously exhausted tumor-specific kills will also reprogrammed by the therapy. So IL-2 variant is mostly acting on both. We think that 33 is mostly acting in the microenvironment, and perhaps in endogenous cells. We know that ST2, the receptor, is not expressing our cells.
Mm-hmm. Thank you.
Anytime, my pleasure. It was a very good questions.
Thank you, Daniel. And so if there's no more questions, then thank you very much, Jesus, and everyone in the audience for joining us today. And yeah, I will follow your research, Jesus, and congratulations. Thanks
Many
for
thanks
joining.
again for the invitation, for the opportunity to share these ideas with you and the way that we are essentially looking at this problem. Let's keep in touch. So enjoy the summer break.
Thank you. Bye-bye.
Anytime, bye-bye.
Okay.